welcome. This is the land of the Noongar people, specifically the Whadjuk. We acknowledge the Noongar people as the first custodians of the land, and I acknowledge their elders, past, present and emerging. We're very privileged in Fremantle to have such a special site. Just out this window is a piece of the original rock, the limestone bar that went across Fremantle before it was opened up as a port, and it was a major gathering point for the local tribes or mobs, and it was really a melting point of bringing together different cultures from the southern area of the urban Darugga to the north. Obviously, there weren't many places that people could cross, so this was one place. The sandbar from Point Walter across to Freshwater Bay, Keens Point, was another one that's known as a Joondalup, and there's other water crossings further along. So it is a very special place, and we think of the long history and legacy that we join. Welcome to the Maritime Museum and part of the West Australian Museum. I'm Simon Carroll, and I have the dubious activity of being the head of science here. And from my point of view, it's a fabulous thing. So I'd like to thank you, the audience, both here and those who will watch our podcast later, the panel members, the staff here at the museum. Philenna has been terrific, and those who have sent in questions for this National Science Week event. Meet the Scientists Q&A. So National Science Week is an annual celebration of science, or STEM as you know it, or some might see STEAM there because arts are a creative and innovative part of the process that we enjoy in Australia, and events large and small are taking place across the nation as we speak, highlighting the opportunities to learn more about the achievements of many people in science and to understand how you can play a role in the future of science, not only in Australia but in the world, because that's what we'll hear today. We're all connected across the planet through the oceans. So this year has been almost living science, as you know. Every day our lives and media are filled with COVID-19. And while we adjust to the regular changes and challenges, there's an opportunity to learn more about virology, immunology, about medical statistics and case loads, about genetic technology and mutations as we track epidemiologically across the outbreaks. So we're living science. I worked on vaccines when I was a postdoc, and so this is back to the future of looking to see how we can develop a vaccine in very rapid time to bring us a different approach to our lives. So National Science Week this year is themed as Deep Blue and Innovations for the Future of Our Oceans. So we thought that we'd bring together 4 panellists who have been working with some innovative tech, looking at jolly deep oceans, as well as their own experience. So we'll have a number of questions after this. But let me just put a little bit of shape into the discussion as to why some of the questions have arrived here today, is that we've, through the Maritime Museum, have partnered with the City of Fremantle Library, and they have a library craft program there. So, you know, Minecraft, library craft, and they are building, they have a competition where they're being asked to build an underwater base. And the builds have to be themed about one of the 3 topics: a research base, which these guys had a mobile one that went down, or a mobile one that went across the top, which is underwater research, shallow and deep water. They need to do a camouflage base, hiding from the monsters of the deep, which you went looking for, I think, and an exploration base, so searching for a new place to live. And so, well, I hope we don't have to do that, although I hear people wanting to call Planet A a lost project and move to Mars, but I think we've got some young scientists in our midst here who are going to try and change the trajectory, I hope. So it seemed a fabulous opportunity to highlight the voyage of discovery that these folks took on the RV Falkor. I don't know whether you've had a look at the video clips of the RV Falkor and the Schmidt Ocean Institute, but we're going to show you them again anyway, because we reckon they're terrific. But I'd just like to let you know the experience that these four scientists had actually was a bit challenging as well, because the vessel left here on March the 8th. And I don't know whether you know, but March the 8th was 1 or 2 days prior to us shutting everything down. So they lived their lives out on the ocean, only communicating by text or FaceTime with their families, while we were trying to adjust to the new normal. And so I can only imagine what it was like not knowing what was going on terra firma, or back on the continent, while they were off in their little cocoon, being safe. They all had to sign up beforehand. At that stage there wasn't any genetic testing, but it must have been a pretty emotionally challenging time. So today we're going to set the scene with some videos. There's 2 of them. One is the video wrap-up from the voyage, and the other one is the short cameos of almost all of the people here about how they came to be a marine scientist. And then we're going to have some questions, okay? The questions will go into the barrel, the fish bowl, you like that? That's good, isn't it? And blue paper, thanks very much for that. It gives all of the elements. Some of these have been sent in ahead from our colleagues at the City of Fremantle Library, and others we've received here, and some have been sent in from people who were planning to come here today. I'm hoping they have. I obviously can't let you know that I know where you've come from, but that's the case. And after the Q&A panel, there'll be an opportunity to talk to these guys with appropriate social distancing, and then we'll wrap it up. So if we could start the first couple of videos, and then I'll refer back. Now, did I do the housekeeping? I don't think I did. The emergency signs sound, beep, 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 beep. You need to go straight out the front and follow the visitor services officer. Toilets are out there and up around the corner, but if you do go, please do be quiet, do it quietly. And thirdly, mobile phones off, which we've already mentioned. So,
I think the thing to keep in mind about this is actually the average ocean depth is nearly 4,000 metres. So the vast area of the world is at that depth or greater. And we've barely begun to really discover what is down there at that depth.
Welcome to the deep sea.
I think for myself, the highlights are literally ever-changing. So the most recent thing we've looked at and found, I'm so thrilled about, I can hardly believe. Then 5 minutes later, the next thing we find, I'm so thrilled about, I can hardly believe we have it.
Visually, we found this incredible solitary hydroid, a relative of corals and sea anemones that stood well over 1 metre tall. The imagery of that, I think, will become the iconic picture for the cruise. I think that's likely to be a new species. So we found some really spectacular worms that are pelagic, that are swimming worms. I'll be sequencing the DNA of them pretty quickly and establishing if they're new records for Western Australia or if they're new species, in which case we'll name them.
Look at it. Yeah, it doesn't have a face. It doesn't have a face. Well, this is what I wanted to see. Oh, it's fantastic.
So high excitement already, even before we can properly sit down. This is a faceless cusk eel. Please enjoy the footage of this amazing creature.
New record for Western Australia. As far as I know, it's the deepest specimen of fish that's ever been taken from Western Australia, and certainly I've never seen any footage of this thing underwater from Australia. So to put all of those things together for this just one thing, let alone all of the other species that we're doing the same thing for, it's just fabulous.
You could imagine maybe 100 years ago you collected something. You didn't imagine a genetic revolution coming. You didn't imagine that it would have this entire new utility, 50 odd years later. And so we're going to imagine what can happen next with our collections. Hello?
Hello. Sorry to interrupt, there's a flashing giant squid on the...
Seriously. It's super exciting, and I actually need to go now. That's cool.
Every moment is kind of new for me, and I live for those. And so it's an incredibly exciting trip because those moments just keep rolling out. They don't stop. Being able to share those unique moments with all the people that are watching is actually really exciting for us as well. So I hope you're as excited as I am. It could be a long day of looking at rocks, or it could be an incredibly exciting day. Either way, you'll have to stay tuned to find out what happens.
In Russia's Glenmore, our curator of fishes to announce that there's a large squid. For me, it captures this cruise perfectly, which is you go out and you're expecting certain things, and all of a sudden you're just taken sideways, and you're now in the moment of discovery, doing something you never expected and seeing something you've never seen. So it's just magic.
I've never done anything like that.
Our goals for this expedition was to understand the biodiversity of Western Australia in a much deeper way. And this is because we haven't actually explored the deep ocean around that coastline very much at all. Simply by carrying out these surveys, we've already increased our knowledge hugely. Every day we're finding amazing critters, new records for Western Australia, new records for Australia, new things that we haven't seen before, and new species. So it's just been an amazing experience.
How did I end up being a marine biologist? So my father was in the Navy, and so I spent many of the years of my youth just exploring the docks, going out fishing with my dad all the time, and that just got me interested gradually, less and less in fishing, but more and more in the organisms. And that at an early age I decided, at 11 I really remember wanting to be a marine biologist, and that carried on through, either through a lack of imagination or it really was something I wanted to do. And that's what I started straight away at university when I was 18.
I guess I was always interested in the ocean and what came out of it. So when I was younger, I would just sort of look along the tide lines to see what the ocean had brought me. And then I got more interested to go and find it first. So I started scuba diving when I was in high school, and then, created a career out of my curiosity for what lives in the ocean. And I just don't see any point in stopping that at all.
For my background, my father was a commercial fisherman, and so was my grandfather. Fishing for scallops, snapper, flounder around Nelson and the top of the northern part of the South Island, New Zealand. Exposed to the sea from a very young age. And it wasn't until university that I started thinking about biological science as a career.
I was always interested in animals right from as young as I can remember. And I loved fish. I used to fish with my dad, and Dad taught me a lot about fish, but I never really thought about being a fish biologist. I was really interested in insects, and when I was 17 I learned to dive, and the first time I stuck my head underneath the water and saw fish in their environment being fish, I was sold. And from that point on I did everything in my schooling and my university to point me right to this point, and I was very lucky with timing that I got the opportunities to take this job.
Straight up The Little Mermaid every time. That's how I became interested in the ocean, was The Little Mermaid, for sure.
Now, in that last video, we heard from Andrew, Nerida, and Glenn over here. Now, this is Lisa Kirkendale, and I've asked her if she'll give a little caption about how she became so passionate about her love of mollusks. And her marine
really
interests.
About the ocean. So I grew up in Canada, and I lived near the ocean and spent summers at my grandparents' home, and I would madly collect things under the rocks, and I'd bring them up, trudging up the hill in a bucket to Grandma's house, and she'd look at them with interest, and then eventually they'd go back. But I think it was that kind of constant exposure. And really, for me, I just couldn't stop asking questions about the ocean. There's just so much to understand and learn that I just continue to ask questions, you know? And so it's really that simple for people that are inquisitive and enjoy challenges. It's just a place that has never-ending questions. Yeah. So
Thank you, Lisa. So I've got some questions. I've got some that I've previously received, and I've got some ones from the door. But let me start with some fish questions. So Glenn is our curator of fishes, and we've got a couple of questions here, Glenn, for you. One's from Taylor, who's 11, and Taylor asks, what are the fish that you see most of, and which are the most rare? So you've got both ends of the spectrum.
Wow.
And let me say that Elliot, who's 13, has asked the question, what type of fish are most common to be seen? So that's a similar side of it. And of course there's, well, you didn't know this, but there's some other questions about fish. What's the biggest fish that's found at the bottom of the ocean so far? So you've got
3 questions.
Trifecta. Well, actually, you've really only got 2.
Fish. Everybody loves
fish,
fish, and I know why, because I do too. Common fish, it really depends where you are. So in the tropics, you'll find on the coral reefs, you're going to get lots of the pretty little fish that you might see in an aquarium. So things like cardinal fish you'll find in big numbers. What else have we got? We got little damselfish. Sorry, I'm trying to think of the common names. I've got to switch my brain out of science mode into common names. And wrasses are really common in those sort of areas. When you get into the deep sea, the sort of areas that we were working in, the fish are really not very common at all. On the way down, though, in the mid-water of the oceans, there's a group of fish called the lanternfish, and they are probably the most numerous fish on the planet anywhere, if you added them all up across all the oceans in the world. And there are many, many species. I can't tell you off the top of my head around the world, but they glow. They have an opportunity to glow, which we might talk about, and I think there's a question later. But they are really, really numerous. In terms of rarity, there are probably lots and lots of fish that we only know from a few specimens, maybe only 1 or 2 specimens. It doesn't necessarily mean that they're rare, particularly when we talk about deep sea. There's a lot of opportunity for these fish to never be seen. So we might just know of only 1 or 2 specimens, but if we found a way to sample down there more reliably, we might find they're not as rare as we thought in the first place. But in terms of things I worked on, perhaps the rarest is probably a blind cave eel. It's a freshwater fish that lives in the caves in the Pilbara, and it's only known from about 11 or 12 specimens. It has a really, really small habitat, so it's a very special fish, but it's a freshwater one.
It's okay. We can desalinate. It's all right. Thank you, Glenn. Andrew, taking on the lanternfish, bioluminescence, midnight zone. I've got a couple of questions on the midnight zone. Maybe you could help us on that. I've just got to find them first of all. Here they go. How are some sea creatures able to produce light in the midnight zone? Perhaps you could explain to the midnight zone to others in a second. And then the second one is, how do organisms that thrive in incredibly low depths survive at the high pressure of these environments, such as in the midnight zone?
Right. So firstly, the midnight zone is a bit of a scientific slang term for the area of the ocean that is below around the 1,000-meter mark is when it officially kind of starts where no light penetrates. So you get what's called the twilight zone, which is above that, which is slightly lighter, only just, and then below that is the midnight zone. So no sunlight penetrates. And so that doesn't mean there's no light, as we suggest. There's bioluminescence, where animals actually are able to produce light. Maybe most, I'm not too sure about percentages, but a lot of animals rely on a special bacteria to actually produce the bioluminescent compounds that flash, such as the anglerfish, the knitted anglerfish there, the little, what's the name? Is it a lappet? The light on an anglerfish? Esker. Inside that, there'll be a colony of bacteria which it gets agitated and glows. So some animals will use it as a lure, such as the anglerfish. Some will use it as a warning signal. Some will use it as distractions. I don't know if the worm actually appeared on the screen, but one of the new species of worms that we've discovered on our trip to Ningaloo has little modified structures on either side of its head, which they will drop off if they're disturbed or threatened by a predator, and much like a lizard dropping its tail, instead of wriggling, they flash. So the worm is called a bomber, and the scientific name actually reflects that. So it's actually officially called, one of the existing species that does this is called the green bomber worm, because it's got little green flashes. So I'm not too sure if that's something that the animal itself does, or whether that's a bacterial symbiont that does it for it.
Oh yeah.
Yeah, still to be worked on. So there's a lot of things that, yeah, like I say, there's a bacteria that do it, and other groups of organisms, the animal is able to do it itself through its own cellular mechanisms, which the biochemistry of which are beyond me.
Thank you.
Can I just jump in about
Yeah.
this? And sorry, it is actually a chemical reaction that these bacteria are doing, and the compound is called luciferin, named after Lucifer, which is another name for the devil, and it mixes with oxygen, so it oxidizes luciferin through, and there's a enzyme that drives that called luciferase, so it's all very fancy, but it is just a chemical reaction that's taking place in these little tiny pits, and it produces a blue-green light because that's the light that travels the furthest in the water. And back to the lanternfish that we're talking about, they're called lanternfish because they have these rows of these bacteria on the belly, and the reason that they have it on the belly, different to the anglerfish, because the anglerfish is using that as a lure to attract things or to be able to see other things. The lanternfish, because they live mid-water, they light up their belly, so if a predator is looking up, they see lights like this against the pale background so that they're camouflaged against the light in the sky.
Thank you. Now, Nerida. Nerida was the chief on that Falkor trip. Nerida and Lisa brought it all together. Someone had to take the lead. The captain's pick was here, I think. And so Nerida was the onboard captain of the team, and so has been around on vessels doing this sort of deep sea ROV mediated science for a fair number of years now, including, sorry, I've got grey in my beard. So I wonder if you might be able to answer some of the deeper sea stuff. So there's one question here that came in from Awesome Girl. I think it's meant to be. We'll call it Awesome Girl. These are names for library craft people. And the first question is, how deep is how much of the ocean has been discovered, and how has anyone been to the Mariana Trench? So 2 questions, breadth and depth.
Okay. So I think it depends a little bit on, what was the word she used, discovered? Yes, you did use. Yep. So it depends what you consider to be discovered, like been there one time, understood about it. And so the way that we kind of estimate our knowledge of the seabed is probably through multi-beam mapping. So if we have a map of it, at least we understand something about it. And so I think in 2017, only 6% of the world's oceans were known through modern mapping techniques. So that's kind of shocking, because since most of our Earth is ocean, so the fact that we knew so little about it was quite disturbing. So they made up this really ambitious project called Seabed 2030, and the goal, of course, is to map everything by then. And so since 2017 to now, we're now at 19%. So that's a huge increase. And so the trip that we did earlier this year contributed, I think, about 11,000 square kilometres of mapping to that project. So we're getting there, but it is certainly a massive amount of effort. So, but at least if we have a map, we can plan dives and visit and explore in a much more kind of knowledgeable way. And the second question was the depth.
Yeah, there's probably 2 parts to the depth. I just discovered another question in here, and this was from Minix in Space, and that is, how deep can a submarine travel? So there's one question, has anyone been to the Mariana Trench? which is a very deep rift in the ocean floor between the Philippines and Papua New Guinea, is it?
Yeah, so directly north of Australia, past Papua New Guinea, but south of Japan. So there's this big sort of semicircle trench at the Mariana Trench, and the Challenger Deep is the very deepest part of that trench. And so I think 13 people now have been to the bottom of that trench, which is kind of amazing, quite a lot in the last 2 years, some as recently as June. So I had to do a little bit of looking myself to understand that and update my own knowledge. So previously we had someone visit it in 1960 in a manned submersible, so in a little submarine. And then it wasn't until 2012 that somebody went back again, and then now in 2019 and 2020. So it's still a place where we know very little about, actually, but we're getting there.
Can I just mention, though? So the question was actually whether anyone on our team has been, and I think that's a no.
Correct.
It is.
So on the bigger, Falkor team.
Okay, that was not the question read out to me, but...
That's okay.
That's right.
But she keeps me honest every day at my work as well.
I do. That is what Shay's Wings asked.
Okay, so I can clarify that one of my colleagues was on the 2012 trip, and so they're a microbiologist, and they were waiting to get the samples of sediment to try and figure out what microbes were growing at that depth. But that's as close as we get. 1 degree of separation.
Anybody want to go?
Sweet. Do we want to go? No. We do. I'm not certain. Should we go back to careers? Are we ready to go to careers? We saw some of them up there, but we know that people think about going into science for some studies that were done at Curtin University in either when they're 5 to 7, there's a light bulb moment, I'm going to be a scientist. I was sitting at a table with a 4-year-old the other day and he said, I'm going to be a paleontologist.
Oh, my son.
And I said, oh, if only I could bring you to the museum and see the collection, because we're actually not able to do that. But your heart goes out. But so in that early zone, the light bulb moment goes on, or later on between about 15 and 25. So the question that is asked here is, when did you know you wanted to be a marine scientist? And you can each answer that very quickly if you like, or 1 or 2 lengthy ones.
20.
20?
Late bloomer.
I started at university as a chemist, so and then almost failed, so I switched.
I was definitely an early person. I poked a sea slug with my Converse sneakers when I was about 5 years old and couldn't believe the purple stuff that was coming out, staining my beautiful sneakers, so it was early.
I'm going to jump in. I grew up in the wheat belt and the ocean was a very long way away. So we only came down to Rockingham for holidays, and in those days there were whaling ships out there that used to do the whaling for Chains Beach, and that was not a very pleasant thing to do. So I didn't really get involved in marine science as a young person.
I think I was probably on the early side as well, but diving certainly cemented that interest in no uncertain terms. But I strayed. So in university, I studied a lot of different things like classics, and I enjoyed literature. I also went in wanting to be an environmental lawyer. I was very determined to save the world. And actually, I wanted more facts. I really wanted something tangible to kind of just wrap my brain around. So I was looking for facts and went back to the ocean.
And I was always going to be a biologist. My youngest memories, I was always going to be a biologist, and when I learnt to dive, I was 17 and that was it. I never had any other option, really. I was sold.
Right, so did you find a megalodon when you were... So there is a question here, I'm struggling to find it at the moment, but someone is asking, do you think we could camouflage ourselves to hide from a megalodon in the deep? And Lisa, you'll probably find it for me.
Yeah, I'll help you.
Yeah, and Thomas, who's aged 10, has asked the question, what is the biggest fish that has been found at the bottom of the ocean so far? So maybe you could quickly work out whether or not, if megalodon was still out there, we could hide from it.
Megalodon's a good question because Ningaloo, we know, because of the fossil record on Cape Range and at Exmouth, we know that megalodons were very common along the Ningaloo coast when they were alive. As to how we would hide from them, I'm really not sure. I think the best thing would be to have an underwater house that looked like a reef. So this is part of the library craft, so building an underwater city. Megalodon, we're not entirely sure. We think they probably ate a lot of large whales, but there's a bit of evidence of seeing bite marks on smaller whales as well of the time, so maybe around 6 foot, maybe around our size. So we certainly would be a small morsel, but I think we would have been within the right size range. So I think staying safely inside a camouflaged house would be a very wise idea.
Right, well that question was from Mrs. Mood. So if you wanted to study a megalodon in the underwater environment for a long time, how would you camouflage your base?
I would make it look like a reef.
And so the biggest fish currently?
Well, it depends. The biggest fish in the world that we know of is the whale shark, but I wouldn't say that lives on the bottom of the ocean.
No.
And it depends how you define the bottom of the ocean, because if you go down to the bottom of the Marianas Trench, is 14, 12, 11. 14 kilometres?
11,000.
11. There's only one fish that we know from there, and that's only about this big. But a true deep sea fish is probably a 6-gill shark, which grows to about 5 metres, and that gets down to about 2,500 metres. So I'd call that a true deep sea fish. Yeah.
So this is a question of judgment, and it comes from Josen Gizo, or Johnson Gizo. I don't know whether the san is without, but anyway. What is the weirdest thing you discovered on the RV Falkor journey?
Well, I actually
anyone can put forward.
Everybody will have a different answer there.
I've brought mine in, which you can have a look at some point. Well, once the questions have started, I'm not too sure about handing it around. It's in a jar of ethanol, but it's a parasite that's related to barnacles, so attached to the bottom of ships. But this one invades other crustaceans such as crabs and shrimp, and feeds off nutrients in the bloodstream. And we picked this guy up quite by, well, not accident. I saw it and we picked it up. We stumbled across it by accident. And it's got little egg sacs dotted all over the legs. So it's, well, I've still been looking into it, but so far it looks like it's the deepest record for this group of parasites. And yeah, so that's my guy. So when you come up at the end of this, you can have a look, and I can tell you a lot more if you can handle it.
More than maybe you ever want.
That's a tough one to beat, team.
You got. Look, I don't think I have a very good weirdo meter. Like, I just think everything is interesting and normal. So I'm the wrong person to ask about that.
You must be able to. What was your most exciting thing you saw?
Well, I think I explained in the video, literally everything was exciting to me.
That's true. And for me too. I was back at the office trying to do work while I'm doing the slow science. Oh, look at that. Anyway, what did you see, Lisa?
So I do think the Teningia dani, which is that octopus squid, the big kind of blood red animal with the flashing lights, was pretty incredible. And probably the most exciting thing was just watching its behavior, how it interacted with the sub, and how it kind of, I don't know, it cloaked itself, like it wrapped itself around, and it kind of did some displaying. So that was amazing to see. I also collected a lot of seagrass at the bottom of the ocean. So at 4,000 meters, I was picking up little tiny pieces of seagrass, and I'm pretty sure the whole team thought, what is she doing? Why is she cleaning up the bottom of the sea? But what we discovered was there were boring, tiny little bivalves that had bored into the seagrass and were living in there and eating off the cellulose that was still present in the kind of the seagrass tissue, and using special bacteria, we think, to digest that cellulose. So it was, cleaning up seagrass off the bottom of the ocean, seeing massive, mighty octopus squid flashing by every day. There was something.
That's fascinating.
Just incredible.
I hadn't heard that story before.
Which one?
The seagrass.
Yes.
Where had that have come from? What depth?
Yeah, so it's, I think, shallow living species of seagrass that are found probably up Ningaloo, and it's been transported to the bottom of the ocean through time. And they're just these little pieces left on the sand, and they're really unmistakable. So just, let's pick one up and have a look. All over to Glenn.
And for me, what I said on the little screen thing there is the faceless cusk. It's just such a bizarre looking fish. Looks like a big tadpole it's only about this long, but its head's the size of a baseball, and it's sort of gelatinous. It's really rubbery and spongy. It has no eyes, and it has really no features on the top of its head at all. So its nostrils are underneath and its mouth is way underneath. So it basically looks like it has no face. Hence its name, but it's just such a weird looking thing, and you saw from my reaction on the screen as well that I really did not expect that we were going to see it here in Western Australia. It's never been recorded from this side of the world at all. So.
Terrific. Thank you. We've got a question at least in scientific processing here, and so there was a question that asked how much of the ocean has been mapped or studied. So in the scientific processing, Megan has asked the question, could you explain the process of sampling such a large area? How would we go about sampling those things? And how often would we collect a DNA sample or a specimen, and what data do we gather on that? So I've paraphrased it a little bit, but it's about what are our scientific methods in trying to catalogue or enumerate the species in such a large area in an underwater environment. It's not like doing a transect in Kings Park or in a 2J or somewhere.
I think Nerida Wilson might like to tackle that question.
I'll start. I have no doubt my colleagues will chime in. I guess one of the biggest challenges of working in the marine environment, and particularly in the deep sea, is that you do have a very limited time. So you might have the ship for 30 days or 3 days. You might have, only so many hours to collect your samples by an ROV. And if you're in a manned submersible, you only have even less time and limited by battery life. So there's always a time constraint on what you can do. And that's really frustrating because we want to, there's always more questions than answers. So you have to really think quite carefully about the questions that you're trying to answer and whether you can actually gather the data needed to answer those questions. So on this particular trip, we had a few different goals. Obviously, we're all biologists, so we really wanted to look at the biology, sample actual specimens. It's very important for our work in taxonomy to have a physical specimen that we can look at in detail and count little legs, take a tissue sample to look at DNA. And so there are things that we really need that sample for. And so it was an unusual trip in having a number of taxonomists behind the screen actually making those decisions. So that was really important to us. But we're not totally selfish, and we realise that there are other disciplines that need that data as well, and working together with other institutions, we can really have more of an impact. So we did a series of video transects, so actually doing quantitative sampling to try and get a baseline where we can compare different parts of the same canyon and then also make comparisons to different canyons, so getting a much broader overview. Obviously the mapping was really important. We were taking water samples to look at environmental DNA. So there was a number of different things we were trying to achieve, but obviously our joy and our passion is about the biology.
Thank you. And so that data, there's another question here from Ghosts, which is Ghost 5. So I think it's Ghosts, but anyway. How do you keep all the data that you collect? How do you manage it? This is a daily question for us at the museum with all of our collections and their data. And how do you store all of this data? Is it online, in books, or anything like that?
Andrew Hosey would like to answer that question.
Thank you, Lisa. So as you can imagine, well, as you've seen, you've got all this video footage and stills as we're sampling. So all of that got kept on hard disks. The ship that we're on has, I don't know how many different sensors, but there's a lot, and there's always pinging and pulling in data. So the ROV data, you would get the data from the sub in a big spreadsheet, and the ROV had 3 different depth measurements, 3 different oxygen measurements. It had its own mapping tools as well as the ship's the vessel's mapping tools for its locality. It had 3 different coordinates for where it was in space behind the boat. So there's a lot of that sort of data, which thankfully we don't used too often. So that's all kept online. The vessel itself manages and curates all of its physical sensors data, so depth, all the weather details that it's picking up as it's going, things like some of the water chemistry and things like that. They all take care of that, and that's all online and actually freely available for those that can make their way through that sort of dense, number-rich data. For us, so specimen here, the general process for us, so as we collected this off the seafloor, we made a little note in the computer that this animal was picked up at this point, which then recorded depth, time, date, physical location, so coordinates, as well as all the chemistry that was being collected at the same time. It got back to the surface. We pulled it out of the bio boxes they're called on the front of the ROV. We took it into the lab, put it on ice to keep it cold, took a tissue sample for DNA and other subsequent like biochemistry type questions that may come up later down the track. Recorded all that, photographed, kept a track of the photograph that's going on. That all came back, well, initially hard copy, into a laptop. As we kept the hard copy, we'll always keep the hard copy. It pays to keep the hard copy. And then this is now sitting in our database. Our databases get uploaded to what's called the Atlas of Living Australia, usually on a 12-monthly, 6-monthly cycle. And so this is now, well, will be, when the next cycle comes around for these guys, this will now be up online with all the data associated with it. And eventually, if this is determined to be a new species, then eventually that data will also get transferred into publication, which will be presumably hard copy and digital as well. And eventually an image of this guy will go up on the ALA website as well. So it all kind of gets taken out into the wild blue yonder, and we try to keep on top of the many thousands of specimens we have coming in on a yearly basis. Any other additional points from the other teams?
No, just that with the Schmidt Ocean Institute that runs the RV Falkor, they have a really strict policy about open access and making data freely available, publicly available. And so that's wonderful. That means that you can go and challenge maybe Andrew's ID of that rhizocephalan parasite on the squat lobster he has over there.
Feel free.
You can look at the data we have, and that's really exciting for us.
And I think it's important to note that while we have all this imagery in national databases and things, the wonderful thing about this is that you can just go to YouTube and actually watch the dives. It's very, very easily accessible. So that will be there in a very easily accessible format.
And if you want to follow up on Andrew's suggestion of the Atlas of Living Australia, you may have seen that. It's at ala.org.au, and you can then go in and have a look at all of the different species that have been observed around Australia. There is a section for citizen science as well, but ALA is at the moment trying to ensure that those citizen science posts are as accurate as possible. And some of these scientists work in that area with fact-checking some of those entries. I've got some questions here about submarines and ROVs. So one question is, how many ROVs have you sent down and what are their names? So I just better work out, I better name who said that question, but they'll know when they see the video because it came from the library group.
So the ROH2D2
Yes.
asked how many underwater drones have you sent down and what were their names?
Yes.
Great. I knew that would come to me. So just to clarify, so there's different kinds of machines and technology that we can send down. So there's AUVs, which are automated or automatic underwater vehicles. There's human-occupied vehicles, so that's what we call a submarine. And there's remotely operated vehicles, and that's what we had on this trip, where we're actually running it from the ship and it's going down and doing what we ask of it. So there's all different kinds of things and machines that we can use to do that work. They do all have names like ships. So the one that we were just using, the ROV, was called Sebastian. I've worked with Tiburon and Jason too, and been in the submarine Alvin. That's my little...
Great. I remember reading about that years ago. So that's 5, is it?
Yeah, there's many. There's some very famous ones, Shinkai. Yeah, there's so many now. It's beyond my capability of reeling them off on the spur of the moment. But yeah, they're dearly beloved to their people.
And I'll just say that there are also very small ones, like this sort of size, that can operate on smaller boats in shallower water. So some of us may have used those, and they don't have names. They just a pet that doesn't have a name.
Actually, in the museum we have a marine scooter, so that you can lie on and it tows you around. So that's, under legislation we have to look at shipwrecks. They're part of the legislation, and so we take one of these out to observe shipwrecks around the coast. So yeah, we've got a bit of kit. But they don't go down to the depth that you need to go to at
I
3-7.
I think it's actually a really good point just to mention that we did go with the RV Falkor and Sebastian because there is no scientifically dedicated submersible like that in Australia. So it's a capacity that's lacking. There is a lot in use commercially and in industry, smaller, larger, but we don't have one dedicated to science. So we brought one in. We called in.
So we're getting close to the end of time. I'd just like to draw the panel's attention to ask them some questions about where they see the next... I nearly let that out, didn't I? The next activity, the big activity that they're hoping to undertake over time, apart from the 21st of November when we open the big place in the city, which we hope will give you all another great thrill of seeing. But so one of the things I asked Nerida to talk about, seeing that I was introducing the concept of genomic testing and COVID, and you might have heard today that the gene sequences of a person in Sydney was different from another person in Sydney and things like that. This is the power of the technology. Nerida, I wonder if you might talk about your Antarctic ice sheet work very briefly.
Sure. I'll just give it a little bit of context, I guess. So with COVID and viruses, they mutate very quickly. So their sequences change all the time. And so that gives us a lot of power to help trace that in a very contemporary time, so over days and weeks and months. When we look at the DNA of animals, we're tending to look at genes that have changes that happen at a much slower rate. So they're giving us a deeper signal of history. And so you can choose particular genes if you know how fast they evolve to answer different questions. And I think what Simon wanted me to talk about briefly was you can also use DNA sequences to try and reconstruct other things. So instead of just trying to understand how closely related animals and biological samples are, I have a project that's looking at samples and populations of animals in different parts of Antarctica. And so we're trying to understand whether historically there was seaways that actually went through Antarctica. And so the history of that connection between those populations gets captured in their genome. And so by sequencing, we can actually inform ice sheet reconstructions and geological work by using biology. So we use geology to help us frame good questions, and sometimes we can help the geologists by using biological signals as well. So sometimes that data can be used in very creative ways, shall we say.
Thank you. Glenn, are you going to tell us about your work in the northwest and your subterranean cave, your eels, or what is your next big?
That's a very good question. There's too many. There's too many to choose from in Western Australia.
Right.
I think perhaps one of the things that we still need to really understand is the freshwater fish in the Kimberley. The Kimberley is such a huge area, and it's so remote and so difficult to access. Some recent work that's done has found that every single river could have a different species in it, even though they might look like they're very similar. When you look at the genetics of them, they're turning out to be that they've been isolated using the techniques that Nerida was just talking about. They've been isolated from each other for so long. They're actually different species living in rivers right next to each other. So there's so many parts of the Kimberley that the only way you can get in is to fly in with a helicopter. And then, of course, trying to sample some of these fish is a challenge when you have a 5-metre crocodile sitting on the bank. So I think there's a huge opportunity for lots of new discoveries in the Kimberley freshwater area.
Thank you. And Lisa, would you like to take the next question?
So some of you may be a little curious about this clam up here. It's not found in the deep sea. It's a shallow water baby giant clam. And that's really been my area of study for decades. So I think some people think that we're all deep sea experts. Well, we're not. So this expedition was my first trip to the deep sea. It was incredible. So I'm very excited for my next step to actually start describing potentially some of the new species that we've found. And that's a long process. I think I'm just going to touch on one question because it was a good one. And somebody asked, what is the most recent, new sea life that's been described, something like that. And so I went and I looked, and it's been listed on Science Alert today, 30 new species discovered in the Galapagos. And so I just wanted to emphasize that, while on our Falkor expedition we might have found up to 30 new species, the process of verifying whether those are new species or maybe new records is long. And that is our core business. That's what we do at museums, and kind of nobody else does that. So I'm excited to start figuring out what animals from Falkor are truly new species. That's me. That's what I'm going to do.
Okay, well, thank you.
November 22nd, I'll be starting right in on that.
And Andrew, you've had an opportunity to give this some thought.
Not long enough. So yeah, as of the 21st, we might have a bit more free time on our hands to complete existing projects. I've got a few under my belt that I need to finally pack away. But the next project that's coming on the horizon is a trip on the CSIRO vessel, the Investigator, which, assuming things kind of ease up in the world with COVID restrictions, is heading out expedition towards Christmas and Cocos Islands. So if you've seen seafloor maps of that part of the Indian Ocean, or to give, I guess, as a bit of a comparison, if you look around Hawaii, you've got a seamount chain, which is little peaks under the sea that drift away, caused by hotspots underneath the Earth's crust. Basically, the mantle's pushing up weaker points in the crust and creating these little volcanoes as they go as it moves along. So between Christmas and Cocos, there's a series of those, not in such a neat little row, but the biology, ecology, and even the geology isn't very well characterized for these areas. We know that they're there thanks to various mapping expeditions throughout the last century, I guess. So this is kind of the first attempt to actually look at the biology of what's on there. And so a team of museums and CSIRO are going out. It's led by the Museum of Victoria. And a couple of us are hopefully going to be on. Like I said, I've got a few things that I've got to get out of the way finished first. And that will be beginning, hopefully, June.
Fabulous. So now it comes to me quickly folding up these answers, these questions, and then asking somebody, a volunteer from amongst us, to come and choose the prize winner. So I'll very quickly... did you want to mention something about why you brought the scrubbing brush?
Yeah, what are these nets
I will
and brushes?
do that while you're folding.
All right.
So I think this, we were asked to bring some things that we enjoy using, and it's not because I like doing dishes. But we had the idea that in the deep sea, the ROV has arms and capturing equipment of a certain size. And if you encounter something that's really big or really fast, it's not so easy to capture it. So I thought maybe we can capture a bit of skin or mucus and get DNA from that. And so I love the contrast of having incredibly expensive and clever technology, holding a kitchen brush and brushing a squid as it goes by, and it actually worked really well, and we have samples from those animals and imagery, and so yeah, it was great because we were never going to be able to capture them. So I love the new and the old together.
You've got some nets.
And I just bought nets. So these are the nets. They're interesting for me because it's the first time that the pilots on board the Falkor that are operating Sebastian, the first time anybody has ever asked them to use a net underwater, which I find remarkable because the skill that these guys have, and they loved it. So these are just standard nets that a diver might use to try and catch prawns in the river. And the guys picked up the skills using these arms. Amazing, and if you get online and you'll see some of the footage that's on there, you'll see them at work. You wouldn't believe that they'd never actually done it before.
I think that brings us to a close, and I'm grateful to my colleagues here for their contribution to the day. I really hope that you've enjoyed it. We will be here for a few minutes afterwards, and as I said, with safe distancing, you could ask some questions. So that's a wrap for National Science Week from the WA Museum Meet the Scientists Q&A. Thank you.
Thank you.